It is Evan, and today we are going to discuss about prions and viroids. First, let us discuss what are prions. Prions are basically these are infectious proteins. PR represents proteins. PR represents proteins. I represents infectious proteins. Infectious proteins, and on represents only. These are only infectious partic proteins which are without genetic material, without infectious material, without genetic material, which is either RNA, DNA, no genetic material, nucleic acid is present. It's the uh, infectious protein, only infectious protein. Now, this is genetic material free, nucleic acid free, infectious protein particle, and that particle is made up of basically wrongly folded or misfolded protein. Wrongly folded or misfolded protein. As it is not having genetic material, you might be wondering how does it cause the infection. We are going to discuss that, but a key to ponder here is that prions cause infection in animals, and prion protein prion protein or PRP is a protein which is present on the surface of our neurons. It is a protein which is present on the surface of our neurons, and that is abundantly found in neurons. Now, whenever there is some sort of infection, whenever there's some sort of uh, a condition in which uh, the gene which synthesizes the prion protein, which is known as PRNP gene, prion PRNP gene, which synthesizes the prion protein, which encodes for the prion protein, right? So, if there's any sort of mutation, or if there's any sort of change in the sequence of the nucleotides of the gene, then it causes to synthesize a misfolded or wrongly folded prion protein. When it comes to its structure is simple, having 29 amino acid with a single disulfide bond. Apart from that, these prion proteins are normally found in neurons, but sometimes if there is an uh, you know misfolded or wrongly folded synthesis of prion protein, then it can lead to infectious prion protein, and at that moment that would be known as an infectious particle prion. How does it actually occurs is that when prion protein is synthesized, that prion, as you can see, that prion is basically a secondary structure protein. It is having a alpha helix structure, so it is a secondary protein. Secondary protein, as well as it is a fibrous protein. Apart from that, uh, these prions basically, uh, normally these are like this is the normal structure of prion, but when they are misfolded, their structure becomes like this. So this is how these are misfolded. So they have infectious, but these are infectious particles because they, even though do not cause any replication, they do not cause any replication within the cell. They do not cause to produce their own copies within the cell, or go and hijack the uh, central dogma and start to synthesize their own protein. So that's not the case at all. They do not have genes, such material, no such thing happens. Actually, what happens is that when these are present, when these are in prions settle within the neurons cell surface membrane, it causes to come in contact with other proteins which are present on the cell surface membrane, and as a result it causes the other proteins to become infectious proteins like it as well. Just like if it is prion, then it will cause the other protein which is in contact with it to act as prion by becoming an abnormal protein, defective protein. So this is how it causes infections. And apart from that, some common diseases caused by it are Kuru, Alzheimer's, mad cow disease, CJD, which is the uh, Creutzfeldt Jakob disease. Creutzfeldt Jakob disease, which is a common example of this. And apart from that, uh, how prion kills. So when prion basically enters within the cell, uh, it causes to It is a slow progress disease. Initially, it does not cause any progress, but once it starts to showcase its symptom, it is adequate to destroy the person uh, within a single month. So this is one thing, and secondly, it is rarely occurring disease, which means in most rare cases this disease can occur, and prions on cell surface allow too much fluid to enter cells, producing a spongy appearance when cross sections of the brain tissue are examined under a microscope. As a result, we are, due to this condition, it leads to formation of several spongy parts of the brain, leading to CJD disease in brainstem, as well as the cerebellum faces a condition of kuru. Kuru basically is a disease. There's a tribe in Indonesia which had the tradition of eating the brain of a dead person during his funeral. So at that moment, this disease of kuru was also, you know uh, in originated from that tribe. So they called this disease in their traditional language as kuru. So at that moment, this was from where the name kuru originated. So these are the affected brain tissue, as you can see in the diagram as well. It's actually a photo. So right, these are some spongy damaged brain tissues, and apart from that, um, this can lead to some symptoms such as fatal insomnia, sleeplessness, Alzheimer's, kuru, mad cow disease, behavioral change, anxiety, and other such symptoms relevant to insomnia, as well as there can be conditions of headache, fever, and yeah, these can be some relevant conditions. So this is a basic intro. Apart from that, when it comes to its treatment, there's no treatment for it actually, but we can have some drugs which can reduce its severity. Apart from that, prevention includes uh, to avoid eating infected animals, as well as avoid eating infected animals, utilizing sterilized surgical instruments, as well as you know it can using sterilized surgical instruments, as well as let me just write that down as well. We can also avoid infected animals, use sterilized instruments. So these are some transmissions of it as well. Transmission, sorry to I forget it to tell that these are caused by eating infected animals which have the prions within their brain, such as cow. Cow is having a disease. A mad cow disease is not caused in humans, but if we are eating a cow with a mad cow disease, so in that case, it can lead to development of prions within our body as well. So um, at that moment, eating infected animals can cause it. Eating infected animals, as well as another condition which can lead to it, is using unsterilized instruments, unsterilized surgical instruments, unsterilized surgical instruments, as well as it can be transmitted, unsterilized surgical instruments, as well as it can be transmitted using during organ transplantation, it can be transmitted. So these are some things which need to be concerned about. And rest we are going to discuss about viroids basically are you know uh, those um, viroids. First and foremost, these were discovered by an American plant pathologist, Theodore or Deer, in the early 1990s, right? So basically, these are supposed to cause diseases within uh, plants only. No cases of animals are reported by viruses. There is a viroid known as hepatitis D virus, but that is not considered as a viroid. We call this as a virusoid because it is having its attack, its infection based over the infection of a prior virus, due to which it is not considered as a viroid but as a virusoid. It can be called as an animal viroid but not as a viroid in actual. Theodore Odier, basically he discovered it, and apart from that, American pathologist was also there, named as when it comes to discovery of prions were discovered by an American pathologist whose name was uh, Pissel, uh, Pisselnier, starting from S. Pisselnier, something like that, S. Pisselnier. So S. Pisselnier was the name. I'm not sure about the actual spelling of it. You can just look into it as well. S. Pisselnier. After that, viroids are basically formed when there is a decapsidization. If there is a ribonucleotide, if there is a ribonucleocapsid, if there is a if there's a capsid inside which there is ribonucleic acid, and if we are performing decapsidization of a ribonucleic capsid, it will cause us to give the ribonucleic acid or the naked RNA genome, and that naked RNA genome will be considered as a viroid. Now, it is not solely a single stranded naked RNA genome. It is basically a single stranded circular naked RNA genome, which is basically also having some double stranded region as well, such that this is a circular region, and within that circular region, some of the region also goes as double stranded region, just like tRNA. In tRNA, we are having some of the regions as double stranded. In the same manner, it is also having some of its region as double stranded. So some of its region are double stranded. Why is that? So that is because it causes to give hydrogen bonding, and hydrogen bonding increases the uh, stability of the viroids. As a result of which, some of the region is double stranded, while other region is single stranded as well. Now, apart from that, when it enters within the cell, it does not cause replication. It does not has gene to synthesize its own protein, but it causes to replicate. Does not cause to synthesize its own protein, but it causes to replicate itself. It has two mechanisms. First includes the replication. It causes to replicate itself. It has a function to replicate. It can replicate itself. It has a function of replication. And second mechanism involves gene silencing. It has a characteristic to silence the gene. How? We know that whenever there's a gene regulation system, over the gene regulation system, let's suppose this is a particular gene. Now, what RNA does is that it basically acts as a micro RNA. It acts as a micro RNA, which has a characteristic to interfere within the ongoing translation process, even though when the mRNA is formed, and when mRNA is formed and it undergoes into the translation process, it can interfere within the translation process and can thus cause the inhibition of the translation process by silencing the gene. Hence, this silencing of gene, which is caused by the interference of RNA, is known as RNA interference. Now, in this case, it can act as it can cause the silencing of gene by attaching with the gene, as a result of which it can lead to act as a microRNA which silence the gene, and this process of interference of formation of protein through RNA is known as RNA interference. So this is how we do it. Apart from that, some common things that whenever it enters the host cell, it basically causes to utilize the RNA polymerase 2 enzyme, because RNA polymerase 2 is responsible for the synthesis of tRNA, as a result of which it also requires something relevant to an enzyme which can synthesize tRNA, because tRNA is also having a double stranded region. In this case, it is also having tRNA. It is also having a requirement of RNA polymerase 2. Apart from that, some common points, that it is having 220 nucleotides. It is the smallest non-plant pathogen. Apart from that, when it comes to the smallest non-virus is sarcovirus, which infects pigs, and that is having around 2000 nucleotides. If I'm not wrong, it is having around 2000 nucleotides, 2000 bases. It is having around 2000 bases, right? So the smallest virus, which is smallest class of group of virus which is known as sarcovirus, which infects pigs, is having around 2000 bases. And these 2000, if there are 2000 bases, which means the nucleotides would be 4000. But here the smallest pathogen in plants is having 220 nucleotides, and it is not having any sort of gene. And apart from that, you know, it's not necessary that there would be a particular region in which there would be a double stranded RNA. There can be any region, right? It depends on the particular type of uh, viroid. So apart from that, some other things I would like to highlight over there, that viroid here has viroid. Viroid basically involves some examples as well. Some serious diseases are caused by it. But before that, let's discuss its transmission. It can be transmitted through pollens as well as seeds, as well as through farming machinery. So how? Through pollens in such a manner that when it enters the cell through the plasmodesmata of the cell wall of the particular plant cell, it then tends to uh, you know uh, remain within the cell. And when the cell produces pollen, that pollen is then utilized. That pollen which is now that from the plasmodesmata, the cell is then supposed to produce pollens. And as it is supposed to produce pollen, that pollen further fuses with the seed, and the pollen further fuses with the ovule of the other plant. It causes to produce a zygote from which embryo arises, from which seed arises, and that seed is then able to transmit that particular viroid which was originally present within the pollen from the plant cells plasmodesmata. So at that case, uh, this is how the viroid is entered within the pollen as well as from the seed. And from the seed, when the seedling will grow, a new plant will be arising, but that plant would be an infected plant, such that it will have symptoms of viroids. Now, coming to this point, that what kind of symptoms can appear. Symptoms which can appear is stunted growth, distorted growth, as well as chlorosis can occur. Stunted growth, distorted growth, chlorosis can occur, mosaic patterns can appear. It's not necessary. Again, it varies from plant to plant. Stunted growth, yellow spots on plants, and mosaic plant. So in this case, this is something which can be affected. Now coming to some of the farming techniques. Of course, when we are farming, then some farming tools also have some sort of seeds and pollens attached towards those farming tools. So when they are used for farming, so during farming, that can also be transmitted. Now coming to some common diseases, speech latent mosaic virus. Speech latent mosaic virus attacks speech, in which there is mosaic pattern appearance over the speech. At that moment, this is known as latent virus, because the symptoms which are appeared are undistinguishable, such that these symptoms appear at the last stage, such that when these symptoms first and foremost do not show any symptom at first, but when they start to show symptoms, they start to degrade and deteriorate the plant completely. Apart from that, when it comes to the other part, which is another example is coconut kadang kadang virus. So here, coconut having a scientific name is Coconus nucifera, and that coconut is basically it causes to form it causes to affect the fruit growth of coconut, because it causes to affect the photosynthetic activity of the coconut virus. Coconut, sorry, not coconut virus. So it basically affects the coconut fruit growth. Coconut cutting virus, which affects the fruit growth, as well as apart from that, there is another virus which is known as viroid, actually a viroid which is known as avocado. uh, it's basically viroid, sorry, avocado sunblotch sunblotch viroid. It basically causes to form several blotches over the avocado in the presence of sunlight. Moreover, other example is spindle potato spindle tuber viroid. Now, potato spindle tuber viroid was the first viroid which was discovered, and it was a first viroid which was observed by Tudor or Deer. Um for and it was the first viroid which was discovered, and that was basically, it is a condition in which we know that a potato is in a tuber shape. It is basically a tuber, but in potato spindle tuber viroid disease, it can cause the tuber of potato to grow in a spindle shape, in an elongated shape, which means its apical growth is increased. Apart from that, uh, there are other such conditions in coconut plant, that leaves of coconut plant also get yellow spots. Leaves of coconut plant, when having the coconut kudunk virus, it's having the affected fruit growth. And how does it occur? It occurs because there are yellow spots present over the leaf of coconut, which leads to chlorosis condition. Now chlorosis condition is occurring, which means that here it causes to do two things: that the gene which is responsible for the synthesis of chlorophyll is not you know synthesizing chlorophyll, which means the gene of the chlorophyll synthesis is gene for the synthesis of chlorophyll is affected. Gene for the synthesis of chlorophyll is silenced by the viroid. Apart from that, there are particular type of proteins present in the coconut plant, which are known as heat shock proteins. Whenever the temperature of plants increase, they tend to create synthesize some new proteins which are known as heat shock proteins, such that when temperature increase, these proteins absorb the amount of heat energy so that the enzymes present within the cell of the plants cannot get denatured. So in that case, when they synthesize the heat shock protein, that synthesis of heat shock protein uh, is also inhibited, such that the gene of the heat shock protein synthesis is silenced by the viroid, as a result of which the coconut plant, of course, forms yellow spots, as well as cannot uh, suffer the heat, due to which their effect is ultimately on the growth of the fruit. Apart from that, these are some basic points I wanted to consider over here. More than 33 types of viroids are now discovered, but there are no cases for animals. Now, this is all about this video. Hopefully, we'll meet again in another video.